day to all. So today's class we will be taking on hemoglobin structure and the synthesis of hemoglobin. So the competency number for today's class is B1 6.11.2, and the SLOs for today's class, or the specific learning objectives today's class is, at the end of the class you should know about the structure of hemoglobin, you should know about the steps involved in the synthesis of hemoglobin. So, going to the structure of hemoglobin, it is a tetrapyrrole. So, this is a pyrrole ring, this is one pyrrole ring. So, there are 4 of these pyrrole rings in the structure. So, these all 4 together join to form a tetrapyrrole. So, this is the pyrrole ring structure. Each of the ring has a number, it is numbered as Roman number 1, 2, 3 and 4. They are joined together by methylene bridges, and these methylene bridges are labeled as alpha, beta, gamma and delta. So, 1, 2, 3, 4 and methylene bridges alpha, beta and gamma. And in the center of the structure you have iron, and in the periphery you have the side chains. The side chains can be of acetyl, methyl, vinyl and propionyl. So, here you can see there is methyl and vinyl. They keep coming for each of the ring of the pyrrole ring, and either they can be symmetrical arrangement or asymmetrical arrangement. Depending on the symmetry, it is called as type 1 or type 3. So, you have the center iron. Iron is present in Fe2 plus form, and this Fe2 plus form has a valency of 6. So, it will be binding to the 4 nitrogen, and the fifth one will be binding to the oxygen molecule, and the 6th bond will be bound to the globulin chain. So, coming to the synthesis of heme synthesis takes place in all the cells, but it is more in certain cells like the bone marrow erythrocytes, and the next highest takes place in the hepatocytes of the liver. This pathway is partly cytosolic, partly mitochondrial. Okay. So, coming to let us go to it step by step. So, in the first step there are 2 molecules which will be combining. You have glycine plus succinyl CoA under the action of the coenzyme PLP, pyridoxal phosphate, which will be the enzyme is ALA synthase. It is going to form ALA. ALA is called as delta amino levulinic acid, and this step takes place in the mitochondria. This enzyme is induced by certain drugs like barbiturates and contraceptive pills. This is important to know because the heme regulation and the disorders due to heme, that is called as porphyrias, are treated by these drugs, or removal of these drugs should be considered. So, the first step, as I had already told, is glycine plus succinyl CoA. They will be joining in under the action of the enzyme ALA synthase. Like if you can see ALA synthase, there is going to be decarboxylation, and the product formed in the first step is delta ALA or amino levulinic acid. So, this is the structure of amino levulinic acid. Okay, after the first step, it moves out of the mitochondria. The first step takes place in the mitochondria. The second step, it needs to move out of the mitochondria. So, once it moves out of the mitochondria, it is 2 molecules of ALA are going to form porphobilinogen. Okay, the enzyme is ALA dehydratase. So, there is going to be loss of 2 water molecules, and it is inhibited by lead. This step is inhibited by lead. Again, this will be important when we talk about anemias and in acquired porphyrias. The coenzyme here is zinc. It is important to know this because in lead, how does lead inhibit the enzyme? Lead removes the zinc, the coenzyme. So, it reduces the action of the enzyme. So, the second step, the first step, now let us go to the second step, 2 ALA molecules, there is loss of 2 water molecules. So, the enzyme is ALA dehydratase, it is going to form the first product, that is second product, that is porphobilinogen. So, as you can see, the ring structure has started to form, the first pyrrole ring has started to form, and make a note that there is an NH2 group. This is important to note that the NH2 group is there because the next step, it is going to be involved in removal of this NH2 group. So, this is the structure of porphobilinogen. You can see the first pyrrole ring has formed and you can see the NH2 group is there. So, just to revise quickly the first step and the second step, then we will go further to the going to the third step. There is going to be formation of uroporphyrinogen 1. It is important that you note that it is now the gens are starting to form. Gens are the porphyrinogen. Gen includes that is going to generate pigments. Okay, so it is going to generate uroporphyrin pigment. This is also called as HMB. HMB is called as hydroxymethane bilane. This is a very short lasting step because it immediately converts into the next one, uroporphyrinogen 3. So, you can see at this step the linear tetrapyrrole. So, you know that porphobilinogen is a first ring structure. The 4 of them are going to join and they are going to form a linear tetrapyrrole. The enzyme is important, you should know this is a very important enzyme for involved in this disorder, that is acute intermittent porphyria. So, as I was telling, so now this is the porphobilinogen, the ring structure. 4 of this ring structure join together to form a linear tetrapyrrole, whereas this is also called as hydroxymethyl bilane, okay, hydroxymethyl bilane, which is going to be immediately converting into a the cyclical structure, that is uroporphyrinogen 3. The enzyme for synthesis of the uroporphyrinogen 3 is the enzyme for synthesis of uroporphyrinogen 3 is the enzyme is uroporphyrinogen 3 co-synthase. Okay, then going to the next step, this is still happening out of the mitochondria, it is taking place in the cytosol. So, after the uroporphyrinogen 3 is synthesized, it is going to form CpG, or also called as coproporphyrinogen 3. And here what happens is the enzyme is UPG3 decarboxylase. So, where is this decarboxylation going to take place? It is going to take place from acetyl, where the carbon dioxide is going to be removed and it is going to form a methyl group, the side chain. We are going to talk about the side chain. So, here as you can see the acetyl, all the acetyl, there are 4 acetyl, the 4 acetyl will get decarboxylated and replaced with a methyl group. And after this step, once coproporphyrinogen 3 is synthesized, it goes back to the mitochondria. So, the first step took place in the mitochondria, the middle steps, the second, third and the fourth step took place in the cytosol. After the formation of coproporphyrinogen, it goes back into the mitochondria. So, in the mitochondria, the next enzyme is coproporphyrinogen oxidase. There is again going to be decarboxylation, where there is going to be conversion of propionyl CoA to methyl CoA, vinyl group, and the product formed is protoporphyrinogen 3. So, you can see the 2 propionates will be converted into vinyl. In the previous step, 4 acetyl were converted, so acetyl group were converted into methyl group. In the next step, the propionyl will getting converted to methyl. So, just again to revise, these are the different steps of heme synthesis. Going further, going to the next step, the protoporphyrin is going to need to be synthesized. The enzyme is protoporphyrinogen oxidase, and in this the methane bridges are getting converted into methyl bridges. So, you can see the bridges are getting synthesized in this step. So, so far the pyrrole ring was formed. Then the side chain got changed from acetyl to methyl, the propionyl to vinyl. Now these methyl bridges, the bridges as I had initially mentioned are alpha, beta and gamma, so and delta. So those bridges are starting to form. They are getting converted from methylene to methinyl bridges. Methinyl is CH. You can see the bond is CH, there are 2 double bond, CH and then single bond. So, again to summarize or to revise, these are what are the different steps, and it is important that you know the enzymes that are involved in each of the steps, because each of the enzyme disorder will lead to a particular type of porphyria. So now coming to the last step where there is synthesis of heme, the protoporphyrin will bind to the ferrous ion or the Fe2 plus form of ion. The enzyme is ferrochelatase. Again it is inhibited by lead. So the second step and the last step are inhibited by lead. And this is again important when we read about anemia and acquired porphyria. So this is the final step where the porphyrin ring has been formed. It joins with the ion and it forms heme. The enzyme is ferrochelatase enzyme. So this is the summary of the whole thing. Let us see it step by step. So the first step, you have glycine plus succinyl CoA forming ALA. In the next step, 2 ALA molecules combine to form porphobilinogen, which is the first ring structure. In the next step, all 4 porphobilinogens join to form uroporphyrinogen 3, where there is enzyme is PBG deaminase. Okay, then as you can see, it forms a linear tetrapyrrole. After that, uroporphyrinogen is going to get converted into coproporphyrinogen, where the side chains start changing acetyl to methyl. The 4 acetyl are getting converted to methyl. There is decarboxylation. So this is how acetyl will be getting converted to methyl. After the formation of coproporphyrinogen, there is going to be protoporphyrinogen 3, where the propionyl is going to get converted to vinyl. So this again to summarize the different steps. So this is how it's taking place in the mitochondria. After the first step, it goes out of the mitochondria. It takes place in the cytosol. Rest of the steps are taking place in the cytosol. After the formation of coproporphyrinogen, it goes back into the mitochondria, and then it continues. It forms protoporphyrinogen 3. Then it goes on to form protoporphyrin, and final step, it joins with iron to form the final product, that is heme. So this is about the structure of, we have so far done the structure of hemoglobin, the synthesis and the steps involved in the synthesis of hemoglobin, heme, and then, so this is the whole summary of the whole pathway which has taken place. And this, if you just write the steps, you can see broadly the steps are taking place partly in the mitochondria, partly in the cytosol. First step in the mitochondria, middle steps in the cytosol, again the final steps again in the mitochondria. Okay, now to the last part of the session, we will be talking about the regulation of heme. How is this heme regulated, how the synthesis is regulated in the body. So the main, there are regulation at the gene level. So at the gene level, it is called as repression or inducer. Here it is mainly repression, where the heme, the product itself goes and acts as a repressor and inhibits the main enzyme, synthesis of the enzyme ALA synthase. Then you even have allosteric inhibition and feedback inhibition. Feedback inhibition, you have, when we had done all the different types of enzyme inhibition in the under the topic enzymes. So in that, you know, the feedback inhibition takes place in a pathway where the end product of the pathway goes and inhibits the key enzyme or the regulatory enzyme. So going on to other type of regulation, you as I had mentioned, the drugs which are inducers like barbiturates, which will induce heme synthesis. They are also, so those patients who are on barbiturates, and if they need to have, if they are having porphyrias, so you need to inhibit the usage of barbiturates. A high glucose concentration is going to prevent the induction. So the high glucose is used for treatment of porphyrias, whereas starvation can worsen a case of a porphyria. So again, so we have done the regulation at the gene level, we have done the regulation at the enzyme level, where it can be allosteric or where there can be feedback regulation, and then coming to the final regulation, the compartmentalization. You know that part of the steps are taking place in the mitochondria, part are taking place in the cytosol, and this is also involved in the regulation pathway because it is regulated, this compartmentalization is involved for controlling the whole pathway. Coming to the final certain small regulatory methods, lead inhibits, you know, the first, the second and the last step, that is ferrochelatase and aldehydatase. Then isoniazid is a drug which is used in the treatment of tuberculosis, that reduces the availability of PLP. PLP is pyridoxal phosphate, which is the coenzyme of the first step, that is ALA synthase. So, that with that we have come to the end of the topic. So, we have done the synthesis of the structure of heme, the different steps it is involved, the different enzymes, the deficiency of different enzymes can lead to porphyrias, and the regulation, how it is, how the synthesis regulated at the gene level, at the enzyme level, and certain inhibitors which will be regulating. Thank you for your attention.